Hello children, welcome to my online video class. Hope you are fine and enjoying my class. So today again we are with the human reproduction, third topic of your NCERT textbook. Under this topic we have selected today the subtopic fertilization and implantation in human. Now once the secondary oocyte released from the ovary, this is what we call ovulation. Ovulation. So during ovulation, one of the two ovary, one at a time. Keep it. It should be a very clear idea you should have that either of the two ovary at a time releases one immature ovum, that is called secondary oocyte, during ovulation, which is taking place almost at the middle of menstrual cycle. Is it very clear? Yes. Now, once the secondary oocyte is released, it will be picked up by the fimbri of oviduct. It is picked up by the fimbri of oviduct. From here, it is coming to the ampulla, and at the juncture of ampulla, this is the isthmus. Here, at the juncture of ampulla isthmus, by that time the ovum is waiting for a sperm to undergo the process of fertilization. What happened during that time? If a sperm is inseminated, through the cervix, it is progressing upward from cervix to the uterus, the sperm tail beating, and with the contraction of the uterine muscle also. So it is moving towards where it is moving? Towards so the sperm here I am showing here is moving towards the ampulla isthmus juncture where the ovum is waiting. Now, when the ovum is waiting and the sperm is reaching to the ampulla isthmus juncture, before that, a very important thing takes place. Immediately after the release of sperm, if the same sperm reaches to the ampulla isthmus juncture approaching to a ovum, it cannot fertilize the ovum. Before it reaches or approaches to ovum, a phenomenon called as capacitation. What is it called? Capacitation. Capacitation takes place. It takes around 7 hours from the release of sperm. Capacitation is nothing but a change induced within the uterus. On what? On the sperm. What happened during the capacitation? As we have already explained earlier, the sperm head is having a cap-like structure made up of acrosome. This acrosome cap contain glycoprotein. What is it? Glycoprotein. This sperm cap made up of acrosome contain glycoprotein and seminal protein. Seminal protein. These two things has to remove from the acrosome in order to fertilize a ovum. This is done when the sperm is entered into the uterus. The uterus induces change by means of which the glycoprotein and the seminal protein of acrosome is totally reduced from the acrosome. Is that clear? Now the sperm is progressing towards ampulla isthmus juncture where the ovum is waiting. So if I rub this part, this is already explained to you what is capacitation. Now the sperm is approaching to a ovum. That ovum is immature ovum, rather to say it is a secondary oocyte. Okay. Now if you recall, the secondary oocyte covered by so many boundaries. What are those boundaries? So let me draw here a secondary oocyte. This is the secondary oocyte. Little bigger size if I draw. This is the secondary oocyte. Secondary oocyte covered by a very tough layer of covering called zona pellucida. This is the layer of zona pellucida, a very tough covering around the ovum. This is the zona pellucida. And another layer of covering, these are all made up of the follicular cells, the granular cells. That another layer of covering another layer of covering another layer of covering this is the corona radiata. Corona radiata. Corona radiata. Corona radiata. Now the sperm which is entering to the ampulla isthmus junction where the oocyte, secondary oocyte is there. This secondary oocyte is covered by this layer of membrane. The sperm has to make an entry into the ovum or secondary oocyte, better to say secondary oocyte, by crossing these two barriers. First barrier immediately is corona radiata. The sperm acrosome, the sperm covered by its sperm, towards the sperm head there is a acrosome. This acrosome contains certain substance, the proteolytic enzymes. So one such enzyme released from the sperm acrosome, that is called hyaluronidase. What is it? These hyaluronidase enzyme secreted from the acrosome helps to penetrate the sperm through the cells of the corona radiata by making a disappearance of these cells. Now the sperm is approaching, so many sperms are there, released, trying to approach to the ovum, Now suppose this sperm has, by means of hyaluronidase enzyme, what is the enzyme? Hyaluronidase enzyme. That enzyme present in the chromosome has already make an entry through the corona radiata by reacting with the corona radiata cells and disappear in the corona radiata cell. Is that clear? Now the sperm is encountering another tough boundary. That tough boundary is the zona pellucida, which is very tough continuous boundary. Now the enzyme, again the enzyme present in the same acrosome of sperm head, covered area, the sperm head is covered by acrosome which contains so many enzymes. One such enzyme, hyaluronidase, helps to penetrate the sperm through the corona radiata. Now another very important enzyme present in the same acrosome called, if I write it here, called acrosin. Acrosin and other some enzyme along with acrosin. Acrosin and other some enzyme which helps to penetrate the sperm through the zona pellucida layer. So zona pellucida layer is also been penetrated. And now the sperm is approaching to the membranous layer around the secondary oocyte through which it has to enter. Now the moment the sperm approaches cross the zona pellucida layer by reacting by hydrolyzing it with the help of acrosin enzyme, now it approaches to the plasma membrane layer of the secondary oocyte, and here a very interesting thing happens. What happened? These sperm now induces a change. What is the change? Within the zona pellucidal layer, once it is making an entry through the zona pellucidal layer, suppose this the sperm has already entered, and now it induces a change. This change now further makes the zona pellucidal layer a strong barrier for the other sperm. So other sperm cannot get a chance. Only one sperm is making an entry. Is that very clear? Once it makes an entry, then after entering the sperm to the secondary oocyte, now the second meiotic division completed. Second meiotic division completes when the sperm make an entry to the secondary cytoplasm of the secondary oocyte, where it will make an entry within the cytoplasm of the secondary oocyte. Now when it make an entry, the second meiotic division completed. As a result of second meiotic division, what is the product form? Product is 1 after second meiotic division, second meiosis, product is ovum or otherwise called ootid and second polar body. Second polar body. Now this second polar body gets degenerated. So second polar body, very small one, and the ovum is much bigger than the second polar body. So this gets degenerated. Now the ovum, which is here at the ampulla isthmus juncture here, that ovum is now here. Now the sperm has already entered, and now the sperm nucleus will fuse with the ovum nucleus to form zygote. What is it called? Now zygote. The process is called fertilization, and by fertilization, that is fusion of ovum nucleus with the sperm nucleus, the resultant product is the formation of zygote. Zygote. Is that very clear? So where the process of fertilization is completed? Within the ampulla isthmus juncture. Now, what will happen after formation of zygote? If I rub this part now, up to zygote, I hope it is very clear to you. Well, fine. So let me proceed further. Let me proceed further after rubbing this part. But before I proceed further, you can see how the zona pellucida layer is being approached by the sperm. So this clipping where we are showing the secondary site here, the zona pellucida layer, the corona radiator layer, the sperms are approaching. Only one such sperm will make an entry. Is that clear? Now, let us proceed. By that time, I will rub this part also, so as to make some space for further discussion. Now, the zygote so formed will undergo repeated mitotic division. Cleavage will take place now. So what will happen after the mitotic division, that is cleavage? The zygote will now form a two-cell stage, 1, 2. Now, this two-cell stage will further divide to form a 4-cell stage. These are all nothing but the embryonic development, and the individual cells here are called blastomere. What is it called? Blastomere. So, 2-cell blastomere, 4-cell blastomere, from here 8-cell blastomere, further division, 8-cell blastomere. So if I draw the blastomere like this, 1, 2, 3, 4, 5, 6, 7, 8. Now it is ready to make an entry towards where? Towards the uterus. But before entering uterus, this 8-cell stage where the blastomeres further divide to form a 16-cell stage. 16-cell stage. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. So at this 16-cell stage, now it is formed by further cleavage. Is it very clear? Now the 8 to 16-cell stage of embryonic development, 5, 6, 7, 8. 8 further division. What is the division? Mitosis. Then forming 16-cell stage. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. These are individual blastomere. So here are 16 blastomere. 16 blastomere. In between each blastomere there are spaces, back-end spaces. So these are, here is 8 cell or 8 blastomeres. Now the 8 to 16 cell blastomere, this embryonic developmental stage is called as morula. Morula. Now, at this 8 to 16 cell stage, the morula will migrate to uterus. Where it will migrate? It will migrate to uterus. And now, the blastomere of morula will undergo repeated division, thereby forming hundreds of such blastomeres by means of repeated division, repeated cleavage. Is that very clear? Well, fine. Now, once such type of numerous blastomeres are formed, then what will happen? This blastomere will arrange themselves in a very nice manner. How? What is the manner? A layer, outer layer comprises of so many blastomeres. These are the individual blastomeres arranged in the form of an outer layer. In the form of an outer layer, a single outer layer. These are the individual blastomere. Individual blastomere. This outer layer of blastomere is called trophoblast. What is it called? Trophoblast. Trophoblast. And the remaining blastomeres form a compact cell inside that is called inner cell mass. Inner cell mass. Is that very clear? So the blastomeres now, when enters to uterus, repeatedly undergo mitotic division, repeated cleavage, thereby forming hundreds of such blastomeres. Is that clear? Now, these blastomeres arrange themselves in one outer continuous layer called as trophoblast and inner clusters of cells called inner cell mass. At this stage, now the larva, that is what we call as embryo or embryonic development, this embryo at this stage is called as blastocyst. 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 Is it very clear? Blastocyst. Now, what will be the fate of this blastocyst? The outer layer of blastocyst, what we call as trophoblast, will now attach with, how it is attaching? I am showing here. Attach with the inner layer of uterus called as endometrium. So see, a part, the blastocyst has already attached with the endometrium. This is the endometrial layer, attached with the endometrium of uterus. Attached with the endometrium layer of uterus. Yes, very clear. And inner cell mass, these are the inner cell mass. These are the inner cell mass. Now, the inner cell mass are having the potentialities to give rise to any kind of cell or tissue. That is, they are stem cell by nature, can give rise to any cell, any tissue, any organ. Now, a cavity develops within the blastocyst called as blastocell. What is that cavity called? Blastocell. So this cavity should develop within the blastomere called blastocell. Blastocell. Is that very clear? Now, the endometrial wall of uterus, that is uterine wall, inner wall, will undergo profuse development, division, proliferation, thereby covering almost the entire growing embryo. As a result, now the embryo is embedded. What is the term I have used? Embedded within the endometrial wall of uterus. This process of embedding the growing embryo within the uterine wall is called implantation. What is it called? Implantation. Is that very clear? Implantation. So when it is implanted, if I show you separate way the covering which is proliferated by the endometrial wall, this is the uterine muscle covering the growing embryo. Now this embryo is called as implanted embryo. Where it is implanted? Within the endometrium of uterus. Is that very clear? Now this embryo will undergo further development. What will happen? The embryo, within the embryo, the blastomeres or individual cells will arrange itself in three different layers. What are the three different layers? One is called ectoderm. Second is called mesoderm. Third is called endoderm. After implantation, now the cells of the blastomere within the blastocyst will further undergo division, repeated cleavage, thousands of cells will produce and they will arrange themselves in three different germinal layers. What is the three different germinal layers? Outermost layer is the ectoderm, middle layer is the mesoderm and inner layer is the endoderm. So, the ectoderm or outermost layer is responsible for formation of the epidermis, the neural system, that is brain and spinal cord, the nasal epithelium, internal ear, formation of pituitary and pineal gland. These are all formed by the ectoderm layer. What I told? Ectoderm layer is responsible for further when it will develop, it will form the epidermis, the neural system, that is brain and spinal cord. It is also responsible for the production of nasal epithelium, responsible for the formation of internal ear, responsible for the formation of pituitary and pineal gland of a fully mature fetus. This is by the ectoderm. What about the middle layer of the mesoderm? Mesoderm is responsible for formation of gastrovascular, the cardiovascular system, heart, blood vessels. It is responsible for formation of the muscles, that is muscular tissues. It is responsible for formation of the mesoderm layer, that is the dermis layer, mesoderm. It is also responsible for formation of the kidney. What about the endodermis? Our enterum, digestive tract, stomach, all the parts are formed from the endodermis. And when these organs will form, by that time the mother will call as a pregnant mother, because of the embryo is now developing within the uterus. So this part we shall take up later on, but before that I shall go the clipping so that you can get an idea about, as I have already explained, the sperm is entering to the secondary oocyte. Then after secondary oocyte, the second meiotic division completed. Now this process of fertilization means the fusion of sperm nucleus with ovum nucleus will take place, resulting in the formation of zygote. Zygote will undergo cleavage to form the two-cell blastomere 1 and 2. Then second mitotic division. All are mitotic division now. The 4-cell blastomere 1, 2, 3, 4. Then next mitotic division is cleavage, 8-cell blastomere, and further division it will form 16-cell blastomere by the time it is enter into the uterus, and the further division of repeated division of the blastomere result in the formation of outer layer of blastomere called trophoblast and inner cell mass. Now the embryonic stage we called as blastocyst. So these stages of development I have described here stage by stage. So if I recapitulate the whole content I have discussed today, the sperm is released, entering to the cervix and trying to enter to the uterus. Within the uterus there is change. What is the change? The sperm cap by means of acrosome, which contain the glycoprotein and seminal protein. These are being dissolved within the uterus by the activity of the uterine fluid. Now the sperm is progressing towards the uterus, and from uterus to the fallopian tube, approaching near the ampulla-isthmus juncture, where the whole event of fertilization. But before fertilization, the sperm acrosome releases the enzyme called hyaluronidase, which helps to penetrate the zona pellucida layer. Hyaluronidase will first help to penetrate or pass through the coronary radiator layer, and another enzyme called acrosine, which will help to penetrate the very top boundary called zona pellucida layer of the secondary site. And now the sperm head is making an entry within the ovum cytoplasm, and immediately after entering to the ovum cytoplasm, it induces a change within the membrane of the secondary oocyte, which prevent the further entry of any more sperm. That is, facilitating only one sperm to enter into the ovum, and now the sperm nucleus will fuse with the ovum nucleus to form a zygote and further development I told. Now one more thing I would like to tell you here, what will be the sex of the growing embryo. That means when the child will deliver, what will be the sex of the newly born child. That is decided immediately after the fertilization. Whether it will be a future male baby or female baby. How it will be decided? See, if you remember about your previous knowledge that female contain XX chromosome along with the autosome, a pair of X chromosome. So when the oogenesis takes place, the ovum so produce all will contain X chromosome. Ovum will contain all X chromosome, whereas the male having one X and one Y chromosome. This is the female we represent in the form of this symbol, and this is the male we represent in the form of this symbol. So while undergoing spermatogenesis, it will produce two types of sperm. One is having X chromosome, other is having Y chromosome. Now, if a sperm, remember very carefully, if a sperm fertilizing an ovum, having the sperm, if it is having X chromosome, it will produce an embryo after fertilization having genetic composition, that is sex chromosome XX. It will give birth a female baby. That means embryo is now developing, which will be would be female baby. So female. Whereas a sperm, if fertilizes a ovum, the sperm having Y chromosome, then the resultant embryos who develop will have a sex chromosome X and Y along with autosome, of course. Then this resultant embryo, which will give birth a baby, that is a male baby. Is that very clear? Up to this we have discussed today. I hope you could get a very clear concept. Remaining part of the embryonic development during pregnancy we shall discuss in my next class. Till then, thank you.